Next, we are going to hear not about single-cell proteomics, but about sub-single-cell proteomics. So a single cell is too much. You can sample only a small part of a cell from an embryo. And Camille Lombard-Banek, or Banek, I'm not sure how you pronounce your last name, from Peter Nemesis' lab is going to tell us about sampling small amounts from embryos, frog embryos, and analyzing the small samples using capillary electrophoresis and mass spectrometry.
All right. Thanks a lot for the invitation and for letting me use your laptop to do my presentation, as I did not bring mine. And so the work I'm presenting today is a method we develop in the Nemesh lab in order to probe embryos as they are developing and live. And so I don't need to convince anyone here of why we want to do a single-cell analysis and to characterize cell-to-cell heterogeneity, how critical that is for many areas. And I'm just going to jump in directly on which area we are specifically focusing on. And this is in the area of embryonic development. And so embryonic cells, the embryo starts as an undifferentiated ball of cell, and then as the embryo develops, those cells start to differentiate one by one. But what is very interesting in the model we use the most in our lab in Xenopus is that even early on, when those cells are not differentiated, they are known to give rise to very specific tissues later in development. And so one of the questions we want to ask is that is those different fates are characterized by molecular heterogeneity in those early embryos. And so here, if I label, for example, that cell, right here, that will give rise to the brain later on in development, you can see that it gives rise to very tight cluster of what we call cell clones. And so this is at the stage 13 embryo, and then here this is at the larval stage. And so you can see how it gives rise to neurotissue type, the retina, the brain, and the spinal cord. And so our goal here is to better understand the mechanism underlying cell differentiation as the embryo develops in a temporal fashion and in spatial fashion, therefore the need for single cell resolution. And the method we use is mass spectrometry. And so we develop, as a slab of lab, new technologies in order to probe and to interrogate the proteome of single embryonic cells. We pick mass spectrometry because it can be a label-free method without the requirement of antibodies, and we can detect thousands or hundreds of thousands of protein at once. And so our early work doing spatial proteomic analysis of the 16-cell embryo demonstrated that those cells actually have different proteomes. And so even though they are not differentiated yet, we can already tell which cell is which based on the proteome, as shown here by the volcano plot. Moreover, we showed that molecular heterogeneity also exists at the metabolome level, and that the differences that we saw are also functional. So when we injected some of the metabolite enriched in, for example, the cell that gives rise to epidermis in a cell that is supposed to give rise to the brain, what we found is that it now gives rise to the epidermis. So that was very interesting. And so similar work can be done at the level of protein to determine whether the difference we saw are functional. But the work we did here was on dissected cell, and I'm going to show how it can be an issue. And so we want to be able to track those single cells as they develop in the embryo, so as they become smaller. And also dissection can be quite low throughput, as you can imagine, and requires quite good skills and good hands. And so we want to find a technique that is not as technically challenging and also can increase the sampling throughput, is scalable to smaller cells, and also leaves the embryo in vivo. So this is what happens to the embryo when you do single cell dissection. I think that everyone can appreciate that your embryo is not very much live and well after the dissection process, and then you end up with here your single cell. So what we developed is a microprobe sampling approach that was recently published in Analytical Chemistry, where we just insert a needle inside the cell of interest. And then after the sampling event, which is just a tiny portion of that cell, we get the embryo still develops and is live and well. However, now we are looking at, as Dr. Slavov mentioned, at a sub-portion of that cell. So we had to kind of accommodate and revise our analytical method in order to accommodate now for that very small amount of sample that we are dealing with. And so this is our overall workflow. So I adapted my lab mate's picture that she had on the metabolomic side for the proteomic side, but pretty much we do everything under a stereo microscope. We have our embryo, our cell of choice, and one of the big advantages of working with Xenopus is that not only the cells are mapped, but also they are very easily identifiable based on pigmentation and the position in the embryos. And so we can very nicely identify here that D11 cell that is supposed to give rise to the brain. And so we sample a small amount of the cell, we release it in, again, like about 5 microliters of digestion solution that we have carefully revised in order to improve the protein identification, the protein digestion, and the protein recovery. And then the method we use to analyze those digests is capillary electrophoresis coupled to high-resolution mass spectrometry. And so because we haven't seen a lot of talk, besides for Professor Ivanov, on capillary electrophoresis, I'll go in detail on exactly what we do for capillary electrophoresis, especially because we build our own system. So we don't use a commercial system, we build our own. And then we identify and quantify protein. In the work I'm presenting here, we did label-free quantification. And so this is the system that, I mean, here I can say I built in the lab, since I was a grad student at the time. Of at least one of them that I built in front of the QX active that was used to generate the data I'm presenting today. And so this is a bird view of the platform, and then here you have what we call the injection platform here where we inject our sample. So this is kind of what it looks like. I have a more detailed view here. And so you have what we have the background electrolyte vial right here, where your capillary resides during the separation, where we apply the high voltage. And here we have our sample loading vial. So we can reproducibly deposit 300 to 500 nanoliters. And by hydrodynamic injection, we typically inject 10 nanoliters of material. So here we're really handling tiny amount of material, which is not that possible with conventional LC method. The ionization system that we use for the work I'm presenting here is the electrokinetic pump sheath flow interface developed in the Dovicki lab. But they have a commercial version, but we actually, again, custom build our own. It's actually pretty easy to assemble, and it's very expensive to buy. So we make our own in the lab, and it's very reproducible. And here, one thing that we pay extreme attention in our lab is to operate our spray in the cojet regime. And this is because it has been shown that this is what generates the most efficient ionization, and so it gives you the better sensitivity for your analysis. And so we have a high-resolution camera that enables us to monitor that we always operate in that regime. So we characterize very carefully our system. And so using a series of angiotensin peptide, we demonstrated that we are at hundreds of deptomole lower limit of detection, as shown here. And then our reproducibility, so if I measure my digest multiple times, I get about 18% reproducibility, and like for the biological reproducibility, so different cell, like the same cell type sampled across different embryos, I get about 25 and about 25% reproducibility. So here this is just comparing the microprobe approach to the dissection, just to show that they were very similar, although we are sampling only a portion of the cell. So as a proof of concept, we validated the approach in the one-cell frog embryo. And so what we did here is that we sampled the animal pole, and then we sampled the vegetal pole. And the reason we picked the one cell is that because there are some known heterogeneity at the mRNA level. And that was something that also was asked by the reviewer that we needed to validate the method, although it was not necessarily known that there would be some protein heterogeneity across those two poles. So what we see here is that using PCA analysis, we had very, very nice separation of the two poles of the embryo. And here I'm highlighting some of the proteins that were statistically and biologically enriched or in the animal or vegetal pole, and also the one that were known to be supposed to be enriched in each other pole. So they're really confirming that our technology, is giving the data that we are supposed to get. And so we went on and applied the technology to now developing cells, and especially we focused on cells that give rise to the brain. And so we identified about 800 protein groups from about 5 nanogram of total protein that were measured. So it's a very tiny portion of the proteome of those cells. And then we had about 450 that were quantified across most of the replicates. And so what I want to show here, and although we had like some comments about correlation before, and so the reason I'm showing those correlation here is that because I collected some of the data a year apart. And so I want to demonstrate here that so two things, that first that the sampling method is extremely reproducible, even years apart, and two, that if you operate capillary electrophoresis with very good standard operating procedure, is very reproducible. And so this is really what is shown here with those high coefficient correlation for the same for different 16 cell measured across a year full span. And so the data that we collected were extremely interesting. So what we saw is that each cell stage first clustered together if we use HEA type of analysis. So again, those analyses are unsupervised. And also one thing that we made sure to control for is that those clustering are also not driven by the order in which we sample the cells. So those numbers here are actually the order in which I sample the cell. And again, those sampling, some of those samplings were done a year apart, and we still got the same clustering. And so here we have the 64. And so what was also interesting is that the 64 and 128 and then 32 and 16 cell were also clustering closer together. And again, that's interesting because 16 and 32 belong to what's called the cleavage stage embryos, and then 64 and 128 are called blastula stage embryos. So they were clustering not only by very narrow time point, but also by the type of global stage that they belong to. And so using also the protein data, we could infer on trends that we saw across those 4 developmental stages. And so we had about 320 proteins that had variable protein amount, and this does not include statistical analysis, so this is just rough trends using for the mean clustering analysis in GPROX. And so what we found about 4 trends of differential expression at the protein level, and then we found 91 of those that were actually statistically significant and evolving as the embryo develop. And so those results are very interesting because bulk analysis of those embryos showed that during the early stage, actually there are no changes. But when you look at those data at the single cell level, now you can really appreciate the small changes that happens during embryonic development and kind of and that the commitment of the cell might actually start much earlier than is previously thought. And so that technology is great again for xenopus, but we also wanted to expand the application to other types of model because not everyone works with xenopus. So what we did is that we expanded the method to zebrafish embryos, and the only change that we really had to make is the geometry of the microprobe. So the microprobe geometry needs to be optimized for the cell or the model you're after. But other than that, everything else pretty much remains the same. And so still from zebrafish, you get way less protein amount than from xenopus. So here the analysis is from about 33 picogram of total protein amount, and we were still able to identify a good 300 protein from those embryos. And so here we only had 3 biological replicates as a proof of concept, but increasing those that n number can maybe start differentiating between the two cells. And here, what I want to also highlight is that the cells here, unlike in Xenopus, are indifferentiable. And so we don't know every time which one we're targeting. And so this shows the need for an increased n number. And I also want to show here that the coefficient correlation, especially between 1 and 3, are not as great as what we showed for Xenopus. So this that might suggest that we could identify eventually some cell heterogeneity also in the zebrafish in early developing embryos. So to conclude, I hope I convinced you that we have developed a method that enables for the in vivo sampling of embryos. And we demonstrated that our approach is valid for getting spatial analysis of one cell and also other stage embryos and also temporal embryos. Here we demonstrated for 16 to 128 cell. And then we also could extend that technology to another model, so the zebrafish embryo, and still identify a good number of proteins. And so our future work will involve, of course, as analytical chemists, we always want to improve the detection limit further. So that's a no-brainer, and also try to eventually sample in the same embryo in different areas, and again see what we get, and if we can get similar data to what we got with cell dissection. With that, I would like to thank my group, so Professor Nemes, as well as our collaborators, Sally Moody and Professor Manzini, who are providing us with embryos and the funding that enabled that work.
Thank you.
Yes, great talk. Thank you. So I have two questions. The first is, I guess you're not able to identify any of the membrane proteins, right?
So we identified a few membrane proteins. We identified a few membrane proteins, but that being said, we did not identify more membrane protein using whole cell dissection than using the microprobe. So in that way, we did not bias compared to dissection.
Okay, so the next question is, how would you control the sampled volume?
That's a very good question. So so one thing that maybe I should have been more specific is that you see here, so to sample the cell and to apply negative pressure to suck out the cytoplasm from the cell, we use a microinjector right here. And so that microinjector, we set it up to a set pressure and then a set amount of time, and that corresponds to a certain volume aspirated from the cell. And so that's how we control the volume.
Well, in my experience, it just gets harder because of the negative pressure. Because when you take out the capillary from inside of the cells, the liquid from the environment also flows in.
So what you can do with the system is that you have an option that is called balance pressure, and so you can set up that balance pressure so that you actually don't have the liquid draw in into your needle.
Thank you. I have a question. First of all, this is amazing. I would have thought it'd be impossible to sample a part of a cell and assume that it was homogeneous. You validated it on a vegetative cell, which is quite large and also not growing, dividing, or signaling. Did you try sampling any of the smaller cells further on in development twice from two different places, just to see if there was a difference?
So when we developed the approach for metabolomics, we actually sampled the cell at 3 different places and kind of measured if we saw like differences in the metabolome, and we did not. That being said, actually, I've not repeated that. Well, actually, I have data that show that, and I put that as a backup slide, so I'm just going to show that. That's going to be easier. And so what we found actually is that we have, compared to whole cell dissection, we have first the same reproducibility, and then we have pretty much equivalent coverage in terms of proteome, if not better, actually than cell dissection. And that alone shows that there is not that much heterogeneity, at least in those cells. In the one cell, yeah, we see heterogeneity, but not in those smaller cells.
Thank you.
I have a question about one of the proteins that you showed as being differentially abundant. That was RPL4. And they have particular affection for ribosomal proteins. So you identified that particular one as being differentially abundant. Are there any other ribosomal proteins that are differentially abundant, or that was an outlier?
No, so actually, that's something also I wanted to discuss with you later, is that ribosomal proteins are actually shown to be differentially enriched in different cell types in embryos. And so this is a fact. And so the protein that I highlight here are proteins that are known to be either supposed to be enriched in the vegetal side or the animal side. And so they are differential enrichment of ribosomal protein, of some ribosomal proteins in differently faded tissues.
Interesting. So we can discuss this over dinner. Thank you. Other questions? All right, let's thank Cam once again.